Hi. So we have learned about the innate immunity in previous session, and now you can understand what kind of innate immune responses are elicited during virus infection. So now I will talk about the innate immune evasion strategies by viruses. Okay. So of course, the pathogen doesn't want to be detected by the host, right? That is quite obvious. They don't want to die. Okay. So they developed some strategy in order to evade the immunity and infect the host. So there are various viral evasion of the innate immune system, various viral evasion strategies, or there are some molecules are there which is used for evasion of innate immunity. So all pathogen, virus including virus, it's not only the viruses have a evasion strategy, all pathogen they have a strategy. Okay, they encode some factor to counteract the induction, signaling and anti-pathogen effector functions. So overall the meaning is that they make some protein, some molecule, some method by which they can evade the host defense mechanism irrespective of innate or adaptive. Okay. So viruses employ the distinct molecule to achieve blockage of antiviral effect of type 1 interferon. So you know that the type 1 interferon is very pivotal in checking the viral replication. Okay. So particularly in innate immunity, right. So this is very important to check the viral replication. Okay. So this type 1 interferon is doing that job. So the virus must have developed some way to evade this interferon, and there are several ways by which they do this thing. They evade the type 1 interferon effects. First is that there is a global inhibition of cellular gene expression. Okay. So there is a global inhibition of cell gene expression. If you remember the type 1 interferon, they basically suppresses the host cellular machinery. Okay. Evasion from innate recognition. Okay. So if so they can make a strategy by which they can not be sensed by pattern recognition receptor. Okay. Inhibition of a molecule involved in a type 1 interferon induction pathway or signaling pathway. So here there are two major ways by which type 1 interferon is produced. One is PRR mediated production of type 1 interferon, and another is the small amount of type 1 interferon which is produced. This is produced, and then this will bind with interferon receptors, and then that will trigger the JAK-STAT pathway, and that JAK-STAT pathway produce a huge amount of type 1 interferon. Okay. So this more amount of type 1 interferon is basically important in checking the viral replication. So viruses have some strategy, or they have some molecule or some protein which they can use it to inhibit this PRR mediated interferon production, as well as they can inhibit the JAK-STAT pathway. Okay. There is a strategy that inactivation of the interferon induced effector molecule. Okay. So you know that once the interferon is produced, then there will be interferon inducible gene. So some viruses have a strategy to inactivate this interferon inducible genes, okay, or effector molecules. Okay. So this is quite busy slide, and here I am just highlighting the cytoplasmic helicases. Here you can see that there is a RIG-I and MDF5. you can see that this RIG-I and MDF5 mediated sensing can be inhibited by, there are so many viruses here. You can see that RSV, MPV, okay, GTOV, June, MAC. There are so many viruses, and they basically inhibit the sensing. Okay, this is true for RIG-I and this is true for MDF5. Okay, there are molecules from viruses which basically inhibit the MAVS or IPS1. Okay, there are molecules which basically inhibit the TBK1 or IKK epsilon. Okay, so like that, there are so many proteins or so many molecules from different viruses which basically inhibit the PRR mediated type 1 interferon production. Okay. Here you can see that there are several viral factor or protein which basically inhibit the TLR mediated innate immune responses or antiviral responses. Here you can see that HCV virus, they have some non-structural protein which basically inhibit this TLR3 mediated signaling pathway. Like that, there are so many molecules. Okay, and all these molecules, the basic aim is to inhibit the production of type 1 interferon. Okay, so and they are acting at different stages. It's not that only there is only one target. So different viruses have a different target. Some targets the receptor, some target adapter, some target some signaling transducer, and some are targeting the transcription factor also. Here you can see that IRF3 is targeted by so many protein from different viruses. Okay, so overall aim of the virus is to inhibit this production of type 1 interferon as well as inflammatory cytokine. Okay, here you can see that there is a JAK-STAT pathway. Okay, here you can see that there is a interferon receptor. Okay, and this interferon receptor is basically is producing after binding with type 1 interferon, it is producing more amount of type 1 interferon. Okay, so here you can see that the receptor itself is targeted by some viral factor, like there is a protein that is here you can see that it is derived from VSV, vaccinia virus. Okay, not VSV, and that is targeting the receptor. Then that is downstream. There are some protein which is targeting the some of the adapter molecule. Okay, or downstream transducer. So there are so many proteins which is basically targeting one or another protein or receptor, adapter, transducer or transcription factor, and overall aim is to shut down the production of innate antiviral state. When I say innate antiviral state, it means that these viruses basically shutting down the pro-inflammatory cytokine production, and they are shutting down the type 1 interferon. Okay, so here I just gave you the glimpse of how the virus or viral factor can evade the antiviral innate immune system. Okay, and just for your note, you need not to remember all these protein. Here just I wanted to show the importance of viral factor, which is basically used to evade the host innate immune responses. So with this I will just complete this session, and in next session we will discuss about the adaptive how the adaptive immunity check the viral infection. Okay, over there mainly I will talk about the antibodies and cytotoxic T cells and Th cells. Okay. With this, I will stop and we will discuss in next session. Thank you.